Hey everyone, welcome to BioBusters. In this video, we'll cover everything you need to know for Subtopic 1.2, Ultrastructure of Cells of Topic 1, which is Cell Biology of the IB Biology HL curriculum. I'll begin by introducing the concept of organelles in cells. We'll also talk about compartmentalization and its advantages, eukaryotes, prokaryotes, and their differences. We'll then move on to the functions of two different cell types, electron microscopes, and identifying organelles in micrographs. Let's get started. First off, what is an organelle? It's a specialized substructure within a cell that serves a specific function. Typically, they're not found in prokaryotic cells, but in eukaryotic cells. With organelles comes compartmentalization. This concept refers to the formation of compartments within the cell by membrane-bound organelles. In eukaryotic cells, the advantages of compartmentalization are that first, there's a greater efficiency of metabolism as the enzymes and substrates are enclosed, therefore they're more concentrated in organelles responsible for specific functions. Second, the internal conditions can be differentiated and optimal conditions for different enzymes are maintained. Third, it allows for the isolation of toxic or damaging substances away from the cytoplasm. And fourth, it allows for the flexibility of changing the numbers and position of organelles within a cell based on the cell's requirements. Eukaryotic cells are very different from prokaryotic cells. One of their major differences is the size of each. While eukaryotic cells range from 10 to 100 micrometers in diameter, prokaryotic cells range from 0.1 to 5 micrometers in diameter. Let's start by looking at eukaryotes. Eukaryotes are organisms with complex cells. They have a nucleus which isolates the genetic material from the cytoplasm, they're compartmentalized by membrane-bound structures that perform specific roles, and they evolved from prokaryotic cells. There are four kingdoms of eukaryotes: Protista, which are unicellular organisms or multicellular organisms without specialized tissue; fungi, which have cell wall made of chitin and obtain nutrition via heterotrophic absorption; plantae, which have cell wall made of cellulose and obtain nutrition autotrophically; and finally animalia, which have no cell wall and obtain nutrition via heterotrophic ingestion. Now let's move on to organelles in both eukaryotes and prokaryotes. First off, we have the plasma membrane. It's a semi-permeable and selective barrier surrounding the cell. It's also a phospholipid bilayer embedded with proteins. Its function is to control the movement of substances in and out of the cell. Next is the cytosol. It's the internal fluid medium of the cell, and it holds all the organelles and contains the enzymes that catalyze the reactions. In eukaryotes, it's the site of some metabolic reactions, while in prokaryotes, it's the site of all metabolic processes. Next, we have ribosomes. They're an organelle made of two subunits of RNA and protein, and they're where protein synthesis takes place. While in eukaryotes, their size is 80S, in prokaryotes, their size is 70S. Now let's look at organelles specifically in eukaryotes. On the left, we see an animal cell, and on the right is a plant cell. Let's identify each organelle. First is the mitochondrion. This is a double membrane structure, and it's the site of aerobic cellular respiration, or ATP production. Next, the nucleus. This is a double membrane structure with pores, and it contains the nucleolus. Its function is to control all the activities of the cell and store DNA as chromatin. The nucleolus is part of the nucleus, and it's the site of ribosome assembly. The smooth endoplasmic reticulum, also known as SER, is a smooth membrane network, and it produces and stores lipids. The rough endoplasmic reticulum, or RER, is a rough membrane network studded with ribosomes. Its function is to transport protein produced by the RER ribosomes to the Golgi apparatus. These proteins are used outside of the cell. The Golgi apparatus is an assembly of vesicles and folded membranes located near the cell membrane. It sorts, stores, processes, modifies, and exports secretory products to be released in Golgi vesicles. Vesicles are small sacs that transport and release substances produced by the cell by fusing with the cell membrane. The vacuole is a fluid-filled internal cavity surrounded by a membrane, and it helps in the storage of substances, and it helps maintain the osmotic balance of the cell and the hydrostatic pressure. Lysosomes and centrioles are organelles found only in animal cells, not in plant cells. Lysosomes are membranous sacs filled with hydrolytic enzymes that work in the destruction of microbes engulfed by white blood cells, in the destruction of old cellular organelles, and in the breakdown of macromolecules. Next, centrioles are paired barrel-shaped organelles located in the cytoplasm of animal cells near the nuclear envelope that work in nuclear division by helping to establish the microtubules. Now there are some organelles only found in plant cells. For instance, the cell wall. This is an external covering made of cellulose that protects the cell, provides support and mechanical strength, so it maintains the shape, and prevents excess water uptake in the cell. Finally, the chloroplast. This is a double membrane structure that contains the pigments used in photosynthesis. Make sure you know how to identify the main organelles in each cell because you may be asked to draw one. Now let's move on to prokaryotes. They are unicellular organisms whose cells lack a nucleus. They have the simplest cell structure with no compartmentalization and no membrane-bound organelles. There are two types of prokaryotic cells: eubacteria, which are traditional bacteria, for example E. coli, and archaebacteria, which live in extreme environments, such as those with high temperatures. Prokaryotes divide using binary fission. This is a form of asexual reproduction. In this process, first comes the duplication of DNA in response to a replication signal beginning at the point of origin. The two DNA copies attach to the membrane and move to opposite ends of the cell. Then the cell elongates. The membrane grows and pinches off, also known as cytokinesis, and the result is two genetically identical cells. The organelles found in prokaryotic cells are the cell wall, plasma membrane, cytosol, pili, flagella, ribosomes, nucleoid, plasmids, and finally a slime capsule. Since we've already gone over the first three, let's start with the pili. They are protein filaments on the cell wall and they help with cell adhesion to surfaces, they aid in the transfer of DNA between two cells, and they mediate bacterial conjugation. Next, the flagella. They are long, slender projections containing a motor protein and they enable the movement of the organism by propelling the cell. The nucleoid is the region of the cytoplasm where all the DNA is found. It controls all the activities of the cell as well as the reproduction of the organism. Plasmids are small autonomous circular DNA molecules that carry a few genes and may be transferred between bacteria. Finally, the slime capsule is a thick polysaccharide layer that protects the cell from drying out. Now let's take a look at two examples of cells. The first are exocrine gland cells of the pancreas. These cells secrete digestive enzymes via the pancreatic duct into the small intestine and aid in food digestion. Their RER synthesizes the enzymes, the Golgi apparatus produces vesicles containing these enzymes, and the vesicles merge with the plasma membrane to release their content into the small intestine. The second example is palisade mesophyll cells of the leaf. These cells contain many chloroplasts for photosynthesis, as palisade mesophyll tissue is positioned right under the upper epidermis, so it's exposed to the highest amount of light. Finally, there's one more thing we need to know: electron microscopes. When compared to light microscopes, these microscopes have a lot of advantages, as they have a much higher range of magnification, so they can detect smaller structures, and a much higher resolution, so they can provide clearer or more detailed images. However, they do have some disadvantages, as they cannot display living specimens in their natural colors, and subcellular structures must be dead before imaging. Now, using the microscope, the following images were taken for different cells. This is a eukaryotic cell of an animal. Pause the video and try to find the different organelles. This is a eukaryotic cell of a plant. Pause the video and try to find the different organelles. Finally, this is a prokaryotic cell. Pause the video and try to find the different organelles. And that's it for Subtopic 1.2 of the IB Biology curriculum. Thanks for watching!